Let's hear it for a great patient stem cell advocate, Ms. Janet Marchibrody.
Thank you very much. It's great to be with you all this afternoon on this very important topic. As Dr. Farshian said, the Alliance for Cell Therapy Now, we're an independent nonprofit organization devoted to advancing safe and effective regenerative therapies. And I have to tell you, we believe that one of the most important steps that we can do in the field to advance this field is to make sure that the science is there. And I know we're going to talk a lot about that today, and to make sure that therapies are both safe and effective. And a lot of change going on. And this goal, the primary goal of our discussion this afternoon, is to help all of you further understand the FDA rules and regulations, and to help you, understand and ask lots of questions about the steps that are needed to assure compliance. So we'll have a lot of good discussion. And let me just sort of walk through how we're going to approach this over the next hour or so. Peter Marks, who's the director of the Center for Biologics Evaluation and Research, or CBER, at the Food and Drug Administration, was kind enough to join us by Skype today. And he's the one who how many of you, by a show of hands, are very familiar with FDA policy? And that's going to help all of us. So Dr. Marks, that was probably about 30% as you're thinking about, and the rest of our panelists. So what he's going to do, he's going to walk through the current rules and regulations associated with cell therapies, and he's also going to share, they've got a lot of really important programs that will help folks figure out where do they fit. Do they need to be, is there approval before you hit the market or not? And so he's going to talk about not only the rules and regulations, but the programs they have in place. And that's going to support the second primary goal of our session, which is to help you to offer insights regarding what are some of the practical steps that need to be taken in order to assess where you are in relation to the regulations and also achieve compliance. And we're so delighted that Dr. Julie Alexson, who's the director of the Regenerative Medicine Clinical Center at the Wake Forest Institute for Regenerative Medicine, she's going to build on Dr. Marks' presentation, sort of talk you through or leverage and build on some practical steps on how you move forward. We're then going to take a short pause. I think some of you probably have questions about the FDA regulations, so we're going to do a little brief Q&A for technical questions and clarification with Dr. Marks on the phone and with Dr. Alexson. And then Anthony Oliva is with us from Longeveron, and he's also a clinical PI and investigator. He's with us here. He's going to sort of take it on the ground. What have they gone through? How have they approached clinical trials and pursuit of an IND? Then we're going to switch gears. And I'm so delighted that Lisa Henning is here with us. She's with the Timmons Group. She's going to talk about, so we'll cover federal policy, and she's going to talk about what's going on here in Florida. What's the current climate in terms of regulation or legislation primarily, and then what to expect in the future. And that's going to be really important because there are a multitude of laws that one needs to comply with. And probably most important is Victor. Victor O'Brien is here with us because this is really all about helping patients with unmet needs, conditions where we haven't been able to address those. And he's going to talk about why these therapies are important to him, his views on, why safety and effectiveness are important. And then finally, Dr. Farshian, we're pleased, is going to wrap us up. We're here at the Academy, and he's going to talk about things that the Academy is doing to help its members its members and all of you move forward on bringing these these therapies to patients. So with that, Dr. Marks, I think we have, and I'll turn it over to Dr. Marks. We should have slides.
Ask you to that end. Okay. Are you able to hear me on your end okay?
Yes, loud and clear. Thank you, Dr. Marks.
Okay. So I'm going to try to keep things relatively simple as I present. And what I'm going to do here, if you go to the next slide, is try to tell you that the bottom line up front here is that FDA is very committed to advancing the development of safe and effective cellular therapies to help address unmet medical needs. And we intend to accomplish that by encouraging sponsor interactions with the agency, deploying all applicable development programs that we have, and also by taking appropriate action when needed to protect patients when there are unsafe products that are marketed. So if you go to the next slide for me. I just want to start with some definitions because this whole class of products that we're talking about, many of them fall under this heading of being human cells, tissues, or cellular or tissue-based products. We abbreviate that HCTPs. And those are articles containing or consisting of human cells or tissues that are intended for implantation, transplantation, infusion, or transfer into a human recipient. And there's a citation to our 21 Code of Regulations there. But examples, just to make it real for you, is they include things like skin, mesenchymal stem cells, hematopoietic progenitor cells from cord blood. And there's a regulatory framework that applies to these, and that regulatory framework was developed to essentially separate out those products which by face value will be taken to be safe and effective when transferred from one individual to another, because as long as reasonable precautions are taken to prevent transmission of infectious diseases, they will do the same thing in the recipient as they did in the donor. And we're not concerned of any kind of safety issue really based on any new use of that. So that the classic example would be that of a skin graft from one individual to another, or a skin graft from one place in the body to another. On the other hand, there are products where they're more manufactured or not than that. They might undergo enzymatic digestion and then culture in an incubator for weeks, and then they're given to another individual. Those have a different set of regulatory questions and need to be separated into a different bin. So as I go to the next slide here, what you see here is the fact that we have these two tiers. So the tier of products that requires significant regulation and is regulated like any other drug are those products which have safety and efficacy that cannot simply be taken at face value from the recipient to the donor. In other words, you're taking something out of one part of the body and perhaps putting it into a different part of the body. And products that are manufactured so that they undergo significant processing, because when you process a product significantly, you can introduce safety concerns because of either chemicals that are added or because the products can change while they're in cell culture. And particularly if you're doing both of those things, using a product in a different part of the body than it started out and having it undergo manufacture, that's when these things, it's very clear that Section 351 of the Public Health Service Act, which is our biologics license provision, which essentially is the equivalent of like regulating a drug, kicks in. And that provision requires a license to distribute a product in interstate commerce. The product has to be safe, pure, and potent, and we have the power to take the product off the market if need be, or recall the product. Now, that's for products where there's a significant manufacturing or significant difference in how it's used in the recipient and the donor. Other products, and if it's an autologous product, that mainly has to, the same things still apply. Those products, though, that don't require that same level of regulation are ones where they're used in a very similar manner in the recipient as the donor, or they're used in a very similar manner in one part of the body versus another part of the body where it's moved to. And if I go to the next slide, please. So that those two tiers are the tiers then that are regulated as drugs, devices, or biological products, section 351 HCTPs, and those that are regulated according to section according to section 361 of the Public Health Service Act. And I neglected on that last slide to mention to you that section 361 is the provision that actually it basically says that FDA is supposed to prevent the transmission of communicable diseases either interstate or introduced into the United States. And things that can be regulated under that authority solely, which don't require pre-market authorization, that simply require registration and listing of the facility with the FDA, those are things that are like skin grafts, they could be bone grafts. Those are things that are simply, they're performing the same function as the recipient, essentially, as in the donor, and there hasn't been significant manufacturing. Again, I'm going to come back to stress this. The important thing here is not to get like crazy about parsing out big differences and little differences, but to really understand that the big concepts here are that 351 HCTPs are ones that are being used for a different function in where they're being placed than they were from where they came from, and they're generally undergo manufacturing that changes them in some way from where they started to where they end up. 361 HCTPs don't have that, those qualities. And we go to the next slide. So we have in our regulations 4 criteria which essentially are the more formal lawyerly ways of saying what I was just trying to tell you. And that is that 361 HCTPs have to meet 4 criteria in order to be regulated as such. In other words, not to require pre-market authorization. That is, they have to be minimally manipulated. Read that for not significantly manufactured. They have to be intended for homologous use only. Read that as performing the same or essentially the same function in the recipient or the recipient site as in the donor or donor site. And they should not be combined with another article. And there's some rare exceptions to that. But what that means is you can't have a combination product there. And either they don't have a systemic effect or have their activity dependent on the metabolic activity of living cells, or if they do have that, they're basically products that are for autologous transplantation. Usually that's the exception for things like stem cell transplants for hematologic diseases. If you go to the next slide. So those are what we have, and we understand that the way I just talked to you about that means that there are a number of things that people have been using where they may have thought they were regulated under 361. That is, they did not require pre-market authorization, that now they may realize that they do have to be under a biologics license application, that is section 351. And what we've tried to do is make it as clear as possible by putting out a suite of regenerative medicine guidance documents, and those are to clarify our existing regulations, to make it simpler for sponsors to determine if they need to obtain pre-market authorization. And we also very much want to expedite the development of safe and effective regenerative medicine therapies, because it's an incredibly promising area. If you go to the next slide. So we have a suite of regenerative medicine guidance documents. The first 2 of these are important because they explain which products simply don't apply to this whole regulatory framework. That's the first guidance, same surgical procedure exception. That explains that for certain things, they don't even fall into this HCTP regulatory framework. An example of that is if you as a surgeon take out the thyroid gland in a total thyroidectomy or near total thyroidectomy, you probably will also take out the parathyroids. You'll then go back and implant one of the parathyroids into the arm, but you probably won't want to throw away the others. So you'll keep those. That's okay. You store those. Even though they're going to be stored, we consider that if they ever get put in later on, that's the same surgical procedure for our intents and purposes, and they don't have to come under this regulatory framework. The second of these guidances is the workhorse guidance, the regulatory considerations for human cell tissues and cellular and tissue-based products, minimum manipulation and homologous use. That explains how we go about determining whether something is significantly manufactured or is being used in a different way in the recipient than the donor. Now, that's, as I've told you, is already in the regulations, but this guidance helps amplify upon that to make it clearer. There's a third device guidance, which helps explain how we're trying to facilitate the regenerative medicine device production and approval. And the fourth of these is an expedited programs for regenerative medicine therapies guidance, which explains this pathway of regenerative medicine advanced therapy designation. And if I have the next slide, I'm just going to take you through a little bit more about a couple of these guidances before we finish up. So the guidance on minimum manipulation and homologous use, in addition to discussing what I've told you about, also describes our compliance and enforcement policy, which was that when these guidances came out in November of 2017, we understood that people would need some time to come into compliance. And because of that, we've given people until November of 2020 to submit an investigational new drug application or marketing application in order to come into compliance. Now, that doesn't mean that we're not taking compliance actions for products that are immediately putting the public health at risk. We will take compliance actions currently, but for those that are of lower risk, we are hoping to get people to come into compliance voluntarily. If we go to the next slide. So that might be described as the stick approach. The carrot approach here is to have these programs for regenerative medicine therapies, which describe FDA's considerations for regenerative medicine advanced therapy designation, and it also describes how we're trying to move the field forward. So the RMAT designation is applicable to products addressing serious conditions for which preliminary clinical evidence indicates the potential to address unmet medical needs. When products get this designation, they're eligible for priority review and accelerated approval. And the accelerated approval provisions have been tailored in this case for regenerative medicine products so that there's a kind of an expanded array of ways to fulfill post-marketing commitments. If you go to the next slide, as of September 30th, 2019, we had granted 44 of these regenerative medicine advanced therapy designations out of 115 requests received. The majority of these are orphan products. Most are cellular therapy products or cell-based gene therapy products. And we continue to receive these products. In some cases, I should say, people don't get their designation on the first try. They come back, we work with them, they get it on a second try. So we continue to work with people in this area. If we go to the next slide, one of the things that we are also trying to do is trying to help people get their products developed. One of the programs we have is the Interact program, which is a informal program where we can give people informal advice on how to go about meeting the regulatory requirements for preclinical, clinical data that might be required for an IND submission. Preclinical data or how to submit a clinical development plan as part of an IND, and what manufacturing information might be required. In addition to the Interact program, we've also recently put in place a program called the Tissue Reference Group Rapid Inquiry Program, or TRIP program. That's a program whereby people who are unsure whether their product is a 361 or 351 product can basically describe their product briefly, usually in just a page or two, and send that to us by email. If you get on our website, we have an email box that goes, and we get people an answer in about a week or less time, explaining whether we believe it's a 361, a 351 product, or something else. If you go to the final slide. So I just want to finish up by saying I've gone through a bunch of stuff. I know Julie will go through things in more detail, but I just want to leave you with the fact that we're very committed to advancing the development of safe and effective cellular therapies to help address unmet medical needs. We want to continue to interact with sponsors to try to move these forward, and we want to work with people using all of our applicable development programs. We're a little bit limited at times because of our staffing. We'd like to be able to do more, but we will do our best, and we are trying to staff up. So we look forward to working with you, and I'll stop there. And I think we're going to take questions after Julie's presentation.
Thank you, Dr. Marks. And so I'd like to introduce Dr. Julie Alexson. She serves as the director of the Regenerative Medicine Clinical Center at the Wake Forest Institute for Regenerative Medicine, and she's going to talk through some of the practical aspects of getting ready.
Thank you. Thanks, Janet. I'm excited to be here and honored to follow Dr. Marks. And I'm hoping to be able to shed a little bit of light on maybe some of the granular aspects of being able to take your products to an IND if it requires that. That's pretty much what we do at the Institute for Regenerative Medicine. We start with the faculty and staff at proof of concept and help to get those products to phase 1 or phase 2 clinical trials before we'd move them to a small company or licensure. So I'm excited to be here, and thank you for inviting me to speak. And so I'll kick off. I think I might have. Sorry. So I'll just tell you just a little bit about the Institute. We have 450 faculty and staff. There's about 50 people in the Regenerative Medicine Clinical Center, all working on different types of applications of regenerative medicine. And that was founded in 2004 by Dr. Tony Atala, who is the director of the Institute. So I just wanted to say, where is regenerative medicine? And it's pretty exciting, because if you looked at this 5 years ago, you would have seen most of those trials in phase 1, and now you're seeing a huge number in phase 2, along with phase 3. So this is done by ARM. They do quarterly reports, but it's very exciting for our field. I actually started out in the field in 1989 in bone marrow transplant. So I've watched it mature throughout that time when there really wasn't regulations that we followed. And then moving into regulations. But this is definitely exciting, maturing, and we're able to treat a lot of patients, which is great. And this just basically shows there's about 1,000 companies in regenerative medicine, which is good, because as we make these products under GMP, we really need the supplies and the reagents that meet those requirements. And so that's really important, because we do a lot of qualification in-house. But as we get the companies, that's going to bring the price down of these reagents and supplies that we have to use. So I do feel that's exciting, because that's one of the roadblocks in our field, is really the cost, and how can we get that down in spec, and you know, whether that's automation or all the different things that have to come together. But that's what we have to work on together as a group. The slides are a little bit off. Okay. So I just want to say is what we look at, our center is the regenerative medicine. It's a generator. So what do? We have a whole clinical center that's going to help to facilitate to push it out to phase 2. At the same time, we're open to collaborative efforts with industry where they can get involved early or any phase of the product development as we move it along. And then certainly after phase 2, typically that would be something that moves to a startup or a licensure of the technology. So this demonstrates our platforms at the Institute, and I would say the most applications are really in the tissue engineered space and tissues. We also have a lot of different cell therapies. We work with perinatal cells, placental cells, muscle progenitor cells, and a few others. We harvest a lot of primary cells that we then expand to make the tissue engineered structure. So say tissue engineered urethra, that's made with the patient's cells. We get a biopsy of the bladder, expand those cells, and seed those onto the scaffold. We also work with a lot of different biomaterials, a lot of different devices, bioreactors and bioprinters, and then some small molecule work. So what am I going to talk about today is how are we getting these therapies to the clinic? these are quite complex, and I'm not sure if some of you work with combination products that would be that complex, but we also do the cell therapy, so we're going to talk really about that. And this just shows what is the Regenerative Medicine Clinical Center. We really start out with looking at the preclinical studies. We can do GLP studies that follow GLP in-house. We have a development team that scales up the product. We make the test articles for the preclinical studies. We have a manufacturing group, it funnels to them, and then we test in-house. We have quality assurance, and then regulatory affairs. So we're the liaison for FDA. We prepare the briefing documents, set up the meetings, submit everything, and help to organize that. So this is one of the most important slides for us in the Regenerative Medicine Clinical Center, because it really tells you step by step. And the lead is really all of the regulations. Otherwise, we're not going to be able to move anything to clinic. It has to be safe. Of course, we want an efficacious product, but we really have to follow through on all these things. And this just kind of shows you, I'm not going to go through all of it, but shows you what we have to do from starting with the animal studies all the way working through LA. So there's a lot of steps and there's a lot of places to work with FDA on that. So as Dr. Marks talked about, the 351 and the 361, and some of you might have a 361 product that just follows good tissue practices, or you might have something that requires the pre-market approval, which would require following good tissue practices and good manufacturing practices. So you're probably thinking, you know, what regulatory path am I in? And I think that Dr. Marks talked a lot about, you know, what they can do to be able to find the right path. And I think that there's a lot of options right now. Like I said, I've been in the field for a long time, and I feel over the last few years, there's been a ton of clarity, a ton of interaction with the FDA, and they're really trying to help everybody get in compliance. So I think once you know what pathways are available, it's going to be easy for you to kind of tap on that and help to push you through to a licensed product. He talked a little bit about the tissue reference group as part of the TRIP. So you can take a document to them, a briefing document, and say, I'm not sure whether I'm a 351 or not, or a 361, and they're going to be able to help you. The TRIP program that's in place right now is in place until the last day of the year, and that's going to give you an efficient result. It's something extra that they're putting in place to really help you. So I feel it would be really good to be able to use this if you're questioning which path I'm in, because it's only going to speed things along. And the problem is if you don't know the path, then when November 2020 comes around, then everybody will kind of be scampering to figure out what path they're in. And then when they start the enforcement, if you're not in the right path, that would kind of close you down and stop you from running, and we really don't want to do that. I mean, what they want to do is see you continue to give your therapies, but to figure out the best way to do that within the regulations. Request for designation is really what center do I belong in? So is it a device, is it a drug, or is it a biologic? Some of you might be using collagen or other things with it. It could be made a combination product. Collagen in certain circumstances would be considered a device if you used it with cells, and it's a combination product. So you may have to ask them. CAT is a new program, and it's just a two-page document that you'd submit to them if you have questions about complex manufacturing. This came up, we had a session on bioprinting and what it takes to really take those to clinic, because nothing's in clinical trials right now with bioprinting, and there's a lot of people working on it. So CAT is one way when you have a complex product to be able to get FDA's feedback rapidly, and I think that that's a very recent program. This just shows you how many different areas you have an opportunity to interact with FDA, which is pretty amazing because this hasn't always been the case. They keep adding programs. They added the Interact program, which did take the place of the pre-IND, but I think it's more comprehensive. And so I think it just helps facilitate your movement into clinic. So if you look at this, there's a lot of steps here that you can use. And if you use those, it's only going to speed up your process because you're going to be more knowledgeable about it. This just talks in detail about all the different programs. So the TRIP, as we talked about, it's an informal, non-binding to give you more information about what path you should be on. The CAT, this talks a little bit about what should be involved in that 2-page document. And all of these things have to be submitted electronically. So FDA does have a process that you go through and what the process is to be able to submit the documentation. Dr. Marks also talked about the Interact program, which is a little bit, it's improved over the pre-IND. Again, it's an informal consultation. And then your pre-IND, which is really going to be your meeting before you finish your definitive studies that's going to take you to first in man. So it's going to look at everything that would go into your IND, your CMC section, your preclinical data, and give you feedback on that, which is really good because if you need additional work to be done, then you'll understand what has to get done. So the other thing I would mention is it's so important that all these documents be clear, because the issue is that you have a certain time with them. So you submit your pre-IND documentation and they'll set up an hour meeting with you. During that hour, you have a chance to talk about questions and issues that you have. If your document's not clear, they're really gonna send you back and say, clarify your document. So you really do want clarity, and if you feel like you don't have that, you need to go to somebody to ask to help with that. But that's the way you're gonna be efficient to move things along. This, I picked out some of the most important guidance documents. These are really important, and I think that recently the FDA has brought many more guidance documents to us for clarity, which has been very important to facilitate our work. So these are some, there's a lot of others. These are important ones that I use on a daily basis. And this is what Dr. Marks talked about basically is really, you're going through here, where does your product fall? Is it a 351? Is it a 361? And then if you're not clear on it, you can ask the FDA. This talks a little bit about what Dr. Marks talked about, what is minimal manipulation. I think there's a lot of clarity here, but that doesn't mean it's going to answer everybody's questions, because I think there's a lot of people with unique products. And so again, I would say, if it's not clear, if you're minimally manipulated, or if it's homologous use, that you speak to the FDA, because there are things that are minimal manipulated, but it's just a few different things. Because if you're dissociating or with an enzyme or chopping or mincing, it's going to be more than minimally manipulated. So you really want to understand that, because you don't want to get in the situation in November where you realize you're not in the right path or not in compliance, because I think that's going to put you behind, and I know you all want to move forward with your therapy. We're excited to be here today to see that happen. So this kind of just talks about all of the definitions. So these are just a couple things I put together. So some tips on, preparing for your IND, because what I think is there's probably a lot of people here that already have data, clinical data that they've collected, and possibly that can be used. As we talk about real world evidence, you could possibly put that together. You may have had a registry already and collected that information. You may have had some preclinical work, or there may be some preclinical work that's been published that's associated with what you do. Those are all options to bring that together, to put that in your package. So I know it looks complicated, and I've gone through a lot of different briefing documents, so it doesn't look complicated to me, but if you put all those things together, there might be opportunities for you to move into the IND depending on what the level of data is that you have. So the FDA is open to looking at your real world evidence or the data that you have. The processing of the manufacturing, that's one thing that's really important. So if you're trying to submit that, is the manufacturing process exactly the same? Because you have to understand, it's at the end of the day, it's the product that you're quantitating, you're making sure it's safe. You're doing the same things you would with a drug to be able to classify that and characterize it. So it would have to be, the manufacturing process would have to be the same. So this just gives a little bit of hints, publications as their data. Important to have a summary table of your data that really clarifies things in your preclinical data as well as your clinical data if you have that. Investigator brochure if it's a multi-center clinical trial. The RMAT pathway. So if you do have clinical data and it's a significant clinical indication, there's an opportunity for you to be on the RMAT pathway, and that could accelerate your technology. When you think about the immunotherapy that's out there, like Novartis, where they were actually able to get a licensure after phase 2. So these are opportunities that you see on the RMAT pathway and other accelerated pathways. The CMC section is very important as it's talking about your product, And then there's another tool out there that FDA has a guidance on, Target Product Profile. And it's really saying, you know, at the end of the day, what are you claiming? What is your product? What do you want it to do? And this document describes that. And again, this helps with the clarity for FDA. So if you add all these things, it's going to make it really easy for them to give you feedback. And not to intimidate you, but these are just some of the things that are important in the IND. And there's templates out there to be able to use, and there's a lot of people that have gone through this process for many, many years. So I know it looks complicated, but honestly, when you get down to writing it, you might be thinking, I have a lot of this stuff already once you do it, because I'm sure that there's a lot of people in the audience that has clinical data already. This is the target product profile that I was talking about. And so this is the briefing document that you would present would have these key sections in it. And then just to mention a little bit about the regulatory considerations for cell therapy as we move along. If you're using allogeneic, you know, we need donor eligibility, safety, sterility, quality control. These products, if it's a 351, are manufactured in a GMP, GTP facility, lot release, looking at purity, potency, and then validating your stability. How long can your product, how long can you use it? The tissue coming in or the sample coming in, how long is it good? So you have to look at all those time points while you're processing your product. And then looking at anything that's added to your product as you're making it. So if there's antibiotics or anything else that you're using, a complete media that might have fetal calf serum, it's not that you can't use it, it just has to be tested for it, appropriately tested, and there's regulations that talk about exactly what needs to be tested. This just talks a little bit about all the different clinical trials there are and how those could be set up, and starting with observational studies, so possibly some of you have done that already. This is one thing that we do. So once we get to a point where the product is finalized and we're going into a phase 1, we go through a complete training with the clinician, the PI, and the whole team that's going to do the surgical procedure. This happens to be tissue engineered corpora, which is in phase 1 studies right now for wounded warriors for reconstruction of penile tissue. But we get in a decellularized scaffold, we take a biopsy of corpora from the patient, we tease out those cells, expand them, and then we put them back onto the scaffold. The scaffold matures in a bioreactor. And so we go through the whole process with the clinician, getting the bioreactor in, determining whether it's going to go in the sterile zone, what's going to go in the sterile zone, who's going to open the bioreactor, who's going to take out the scaffold, and then he or she gets to cut it, put sutures through it just to understand what it will be like before he gets into surgery. So we go through a complete training exercise before that. This was a special report that was published by Peter Marks and Scott Gottlieb in 2018. And I think it just gives us another perspective on how could things be done. Usually you have a single center that might have several different trial sites with a single product. But in this publication, they suggested that possibly you do the development together at several different facilities, and it goes through product development, and you'd have the opportunity to apply for a BLA, each one of you would. So it's just a different way of thinking about it, but sharing that knowledge and work in the beginning together through a group, because it can be expensive as you go through development. So I think this is a great option to be able to use this in clinical trials. Another thing I just wanted to mention is OTAT has a lot of webinars, and so you can actually just go right online with FDA and be able to see, how do I prepare an IND, what happens in a pre-IND. It has a lot of different webinars, and I think it can be quite informative if you're really interested in understanding a little bit more about the regulations yourself. So a lot of people ask me, how long does it get from proof of concept to clinical trials? It's a hard question. One of the toughest questions. It could be 7 years, it could be 12, it could be 15, but it really depends on how organized you are, and that's really the message that I wanted to be able to present today, is that if you're to take advantage of all the FDA programs that they have in place, they're at most of the meetings, just like Dr. Marks was, so kind to be at this meeting to be able to present and give us his viewpoint on the regulations. And so we have a lot of interaction with them right now, and also with these webinars. So I think that there's a lot out there for us and a lot of ways that we can be compliant. So I think we just have to ask and reach out. And so I appreciate your time. Thank you so much.
Stay up here. Thank you, Julie. I think we should, so we've covered a lot of, if you'll stay up here, that would be terrific. We've covered a lot of technical ground, FDA rules and regulations. It probably feels overwhelming to folks. And I thought before we move forward, let's pause a minute and have some discussion or questions that you might have for Dr. Marks or Dr. Alexson. You know, what challenges are you experiencing? You know, no question, all questions make sense. This is such a great opportunity to get feedback, informal feedback. And I have one gentleman here in the front. Should we put the mic around? This one? Okay.
Let me give you a moment to think about what I'm asking. Many physicians and other practitioners are concerned about November of 2020, that what is it that legitimizes a physician to use stem cells in biologic products? That's been a big question of mine, because there's been certain organizations that say that you, as a solo practitioner, you need to be involved in an IRB to be able to do this kind of work after November of 2020. And it seems like an onerous thing to do for a solo practitioner, unless you're involved with a manufacturer of a biologic that is involved with an IRB. And my question is, if you are involved with a manufacturer, distributor of biologics who has an IRB, does that make you legitimate after November of 2020? Or does that make you, well, I don't know, adequately trained and adequately accepted as?
So this is Peter. Do you want me to take a stab at answering that?
Yes, Peter.
Go ahead.
Okay. So, and I'm hoping you're hearing me okay. I think that the issue here, there's a lot to unpack there, but I think the idea would be that if you're working with a product that is not accepted because it's a 361 product after November 2020, you would need to be associated somehow with conducting the work under a informed consent and under the auspices of an IRB. If you're working with a sponsor, they're generally going to have the access to helping you figure out what IRB an IRB that you're going to have to access. It could even be a central IRB as is preferred. And then you'll be getting appropriate consent from patients. The actual, how you actually qualify practitioners to do this, that's not FDA's business. That's state boards of medicine, but the products themselves and how they're used, that's what that will require at that point. For products that would fall under our regulations, one would need to be, if it is a 351 biologic and it is being used in an investigational setting, it would need to be come under an institutional review board's oversight. But you as a physician, the ideal way, I think that, and this is one of the ways we're trying to help make this happen, the ideal way that this would happen would be you would become involved in either a collaborative trial or a multi-center trial under a sponsor, so that the data are generated. And the sooner the data are generated and submitted to the agency, the sooner a product would be approved, and then you would actually have something that could be marketed.
I have a follow-up question, please. Is it safe to say that in November of 2020 that there will be some companies who don't meet FDA criteria and will have to close?
I think it's safe to say that that's probably going to be the case. It's that they will either, ideally, they would actually just close up shop because rather than violating our regulations, but if necessary, after that time, if they continue to operate, we will take enforcement and compliance action as appropriate. Now, I will tell you, our goal is not to be there with a big stick. Our goal is to work with people. So, if somebody came into us a few weeks before and said, hey, we're in the process of getting ready to file our IND. Here's a draft, but it's not really ready. We're not going to run on November 20th of 2020 and cite them. But I think that those who are going about thinking that this is just going to go away, it's a problem. And let's just think about this as scientists and as doctors. The issue here is not that we're trying to be. We're not trying to be like rules focused. We're trying to be science and clinical medicine focused, which is that we need to get the data that actually show that these products work. And unfortunately, one can't just simply rely on anecdotal evidence. I'm a hematologist oncologist by training. I was actually involved when I was a fellow and junior attending in breast cancer bone marrow transplants at that time for metastatic breast cancer. And as we learned, even though at that time, I'll be honest, I was convinced it's had to be helping women. At the end of the day, we found out that it wasn't, and it was probably hurting them. So we need the data. And the goal here of these regulations it's not to make people's lives difficult. It's to help us have products that actually are supported by science.
Thank you, Dr. Marks. I know I'm watching the time. Maybe we're going to have Q&A at the end as well, but I wanted any questions.
I'm going to stay on till the end, so I will be here at the end as well.
Oh, that's terrific. Thank you. Any other questions related to regulations? We've got, I think, Dr. Farshian, we can take 2 more questions and then we'll... Okay, all right, let's move. So thank you, Dr. Alexson. Our next speaker is Anthony Oliva. He's, as I mentioned earlier, he's with Longeveron. He's also a senior scientist and PI overseeing scientific and clinical studies, and he's going to share some experiences on the ground. So you've heard a lot about the rules and regulations, but his on-the-ground experiences with their clinical programs, manufacturing, and some regulatory aspects. So please join me in welcoming Dr. Oliva.
Can you hear me? Is this on? Do I need the microphone? Yes. Okay. Did it go out? Okay. All right. So I'm going to be a little less heavy on the regulatory aspects. I think you've gotten beat to death with that already. So I'm Dr. Oliva. Just for disclosures, I work with Longeveron. We're an academic spinoff from the University of Miami. We're a stem cell therapy company. And my disclaimer, these are my own views, not the company's necessarily. Okay. So as the name applies for a company, we're interested in aging-related disorders. Okay. And so there's a few things on this. Expectancy has gotten a lot better for us. So, we're living about 80 years old on average in the US, okay? But there's a problem with this, which is our health span, our healthy years of living have lagged that, okay? So right now in the US, we can expect to live about 11 years in a pretty debilitated condition, okay? On average, and that's only getting worse as the life expectancy or the life expectancy is going up. Okay, so for us, it's this comes down to the aging process in general, and how can we go about treating the aging process in order to overcome these aging-related disorders? Because almost all these disorders, the root of them is the aging process itself. Okay. And there's a lot of common pathology in this, and I won't go over this for sake of time too much, but things we hear a lot about, fibrosis, inflammation is one of those key aspects leading to a lot of debilitation with regards to aging, degeneration. I feel like this keeps going out on me. And another thing is decrease in stem cells themselves, okay? So just to give you, I think the battery is going out on this. Mesenchymal.
We can hear you though.
Yeah, is that okay? It's it just keeps like going out on me though. Okay. He's from LA. So you get the idea. When you're a newborn, you got a lot of stem cells. As you get older into your 80s, you have a lot fewer of them. Okay, and I'm a neuroscientist by training. Okay, and one of the things I'm really interested in is how do you treat a condition like this? So here's your healthy brain, and here's your brain on Alzheimer's disease. Okay. This is pretty complex pathology, and this is the type of situation where a simple, small molecule intervention, it's not going to work. Okay, you need something else. And to me, the way, if you're actually going to fix this, is to use the stem cell therapeutic approach. And the reason for this, because stem cells, and in particular ones I'll talk about as well, they have these pleiotropic mechanisms of action. They can do a lot of things at once to help solve that pathology. Okay. And in our company, we produce, one of our products is Allergenic mesenchymal stem cells. Okay. And again, the reason for this is multiple mechanisms of action, these pleiotropic mechanisms of action. Okay. So in terms of us, what we do is we, one of our products, Regenerative mesenchymal stem cells, these come from bone marrow of young, healthy adult donors, highly screened to be free of communicable diseases, metabolic disorders, et cetera. Very stringent screening process. And these are not minimally manipulated. We get them, we culture expand them to high purity. They go under high QC and quality assurances to make sure that the product in the end being given to those patients is a safe product and a well-characterized product and a reproducible product, okay? Because what we want to do is actually bring something to market that anyone can use in the end. So we have a number of clinical trials currently running in the U.S. and see it went out, and a registry trial going on in Bahamas. So in terms of this, it's a small company. We've only been around for a few years now, and I actually jumped at academia to join this as a startup because, stem cell therapy, aging-related indications, this is the way you're going to do it. So I've been there ever since. But for us, we have had a lot of very good interactions with FDA. Okay, they have, I wouldn't say have gone completely out of their way, but they have very much been advocates for us rather than antagonists. They've tried to get us over the finish line. We have INDs in place for all of these studies. Okay, we haven't had clinical holds because they've helped us. They've been straightforward. If you want to not have a clinical hold, you need to do this. Okay, we do it. I mean, that's it. So, for us, it's been very good. And we, and the feedback we get from them helps guide our clinical programs forward as well. Okay, so, in terms of, sort of the bigger picture, some of the stuff that, Dr. Marks was talking about, as well as Dr. Alexson, it's getting from the anecdotal evidence to the actual, hard science behind whether or not these products are working. And that's exactly what we're trying to do here. We have a number of clinical trials in phase 1, okay, which is, those are safety trials, okay? For this? Is that working? Yeah, this microphone I don't think is on.
You gotta push it
fine.
You can hear it? Oh, okay. I can hear myself echoing back here. Okay, it's a lot different. Okay, so, in terms of this, phase 1, safety trials. Okay, this is to make sure that your product for that indication isn't going to harm the patients. Okay, we do have a couple of trials and safety studies. We have an Alzheimer's disease trial for one of them that we just finished enrollment on. We have excellent safety data on it. We're already preparing to put in our phase 2 trial. The phase 2, these would be early effect trials, okay? So your primary endpoint here isn't just safety. It really is, are you seeing a clinical benefit, okay? Are you seeing a change in biomarkers? Are you seeing improvement in a clinical outcome, a clinical surrogate? For us, one of the things that's important is that, for these, we're interested in aging-related indications, okay? We don't have 60 years to wait to see, okay, well, they lived for 60 years with a lot less debilitation going on. We don't have that. So we have to work with surrogate markers, things that are, potential ways to, these are, ways that are potentially indicative of those long-term clinical benefits. So with something like aging frailty, decreases in hospitalizations and falls and fractures. Okay, so with FDA's guidance, they have helped us, with this. If you want to be able to, get this to a BLA, you're going to have surrogate markers go along with it. So something that's a patient-reported outcome, something that's a some type of physical function outcome, as well as a biomarker to go along with it. Okay, and for us as a company, we have treated over 300 patients so far. Okay, we've had no treatment stop prematurely. We've had no adverse events or serious adverse events related to our product. Okay, and that goes kind of along with what most people in this room know, which is MSCs have a very high safety profile, but we're showing it in a very rigorous way. Okay. Okay, so I'm not going to go over all the trials in detail, but I'll give you just a high-level picture. Aging-related disorders, okay? A lot of signs and symptoms of this: diminished cognition, cardiovascular status, immunosenescence, which is run down to the immune system as you get older, pro-inflammatory state, diminished strength, diminished endurance capacity, decreased stem cell activity, degeneration. And from our clinical studies so far, we've had good indications of the ability of allergenic MSCs to improve all these things. Okay, so improve cognitive function, okay, which is actually, that has springboarded into our dementia program. Improve cardiovascular status, improve lung function. Those are coming from our aging frailty trials, and all this stuff is on clinicaltrials.gov. Improved immune system and decreased inflammation, and improve musculoskeletal function. Okay, so if you are not something like our company where you have your own GMP facility, okay, and you're doing everything by the book, okay, but you do want to run a clinical trial, okay, then you're going to have to team up with a manufacturer, whether it's us, whether it's Dr. Tala's lab and Julie, or whoever it is, okay. There are ways that you can accelerate getting over that finish line, okay. Putting in the IND, you don't have to have your own manufacturing section. You can cross-reference or get a basically letter cross-reference to someone that is manufacturing, and you're using their product, okay. That product has already been vetted by FDA, okay, and that's where you want to be. You want to have someone that is making something under IND or under a BLA or a CMF, something where it's a quality product and what's being put in your patients. It's it's something that will be under high stringency, high QA, QC release criteria, and that's what you can use. So from that, you can build your clinical protocol, your informed consent forms, and put together the IND that then will get past an IRB, and you can go ahead and continue with your studies. Okay, so there's a lot of ability to do that. But what you do have to team up with is someone that's, being in Miami, not, working out of a van in Hialeah, okay, which we have some questionable characters, not necessarily in the stem cell field, but, in medicine that do some questionable things here in South Florida sometimes. Those are the bad characters. You want to team up with good characters that actually is going to let you get past the regulatory agencies. Okay, and for us, we do everything by the book. We all our domestic stuff goes through FDA. For Bahamas, we work with the Bahamas Ministry of Health to run those programs. We work with the Japanese PMDA, and we're going to be getting trials going on in there as well, and elsewhere throughout the world. Okay. So one other thing is, when you're doing these studies, trying to conduct these highly rigorous studies, you can go ahead and go after grant money for it. Okay, so that's another thing, and have someone else help pay for it, because you are actually trying to get to an answer of something that can lead to a product in the end to be available to everyone. So this is my last slide, but I am going to thank all the people, because I've gotten a lot of grants for all of our studies have been grant funded, fortunately, so we haven't had to use our private funding and dilute that out. And for us, this is a really important thing, because every time you can get a grant, that's another investor you're not trying to find. That is not someone in the end you have to pay off, making for a yet more expensive product, because you're paying off all this initial investor. So this is undiluted funding for the company. So 2 grants from NIH supporting our aging frailty and a metabolic syndrome trial, Alzheimer's disease, 2 competitive grants from them supporting the entire Alzheimer's program. It's been great. And then we are a South Florida company, but we have 2 grants from the state of Maryland under this Maryland TEDCO program. We were, I think, the only ones, they were the first ones to get one of these that was out of state, and we got 2. So we're really happy with state of Maryland right now. But actually, this is a good win for them too, because it's opened up other business in their area. And I'll just keep it there, and I'll leave it for questions later.
Dr. Oliva, and now we're going to switch gears. We've spent a lot of time on federal policy, rules, and regulations, and I'm so delighted that Lisa Henning is with us. She's with the Timmons Group, and she's going to talk some of the current and maybe what might be on the horizon when it comes to Florida law. And let's use that one. That was a better. She's going to get mic'd up here for a sec. And hold your questions. I know there are lots of questions. We've heard a lot, and we're going to hold those till the end. Great. Thank you, Lisa.
Thank you. Well, hello, everyone. I am going to talk to you about nothing that has to do anything with science. Unless you're potentially a psychiatrist asking why anyone would work within the governmental process. I am what is often referred to as a dirty scum-sucking lobbyist. So, that being said, I got to proudly go home and tell my mom one day that that was what I was going to be when I grew up. And I did that literally at the age of 12. At the age of 18, I was already done with my initial bout of college and started with Governor Martinez in his administration in the state of Florida. So I was very young and thought I knew everything, and very quickly, within 4 years, became a lobbyist. And one of only 5 female lobbyists in the state of Florida at that time. We now have over 3,000, but at the time there were only 5 of us, and I was a child. So, that being said, that lets you know me. Where I want to let you go is to tell you where we in the state of Florida are currently sitting. We are sitting right smack dab in the middle of the Senate President and the Speaker of the House's tenure as the governing bodies. So we are 2 months away from their final session. What you're going to see is a lot of jockeying about 4 months from now for everyone to go with the new and incoming Senate President and Speaker of the House. But currently we have a fabulous Speaker of the House named Speaker Oliva. Speaker Oliva is very, very proud of and encouraging of stem cell research and stem cell therapy. He likes it. He does not want it strangled. So, that being said, I got a lot of help from Speaker Oliva and some chairmen in the health policy committees to make sure that legislation that was filed last session that would have restricted this stem cell research and usage only to hospitals and universities. So that being said, that was me last year. I'm very unpopular with some people, but very popular with the stem cell community. Okay, the American Academy of Stem Cell Physicians, okay, their priority and why they have me is because they want to continue safe and adequate use of stem cells. No one side or the other. Very, very focused on continuing the use with minimal restrictions but good safety standards. So in obtaining that goal, they hired Timmons Consulting, which consists of 3 lobbyists. There are 3 of us. We work with many groups, but most specifically, we are going to be working with a gentleman that I'm going to be introducing you guys to in a little bit via pictures. But we currently have been working with the manufacturers, lobbyists, to come up with legislation that would put some safety standards in place in Florida, because Florida is kind of, we were referred to as the wild, wild west of stem cells. So that being said, Senate Bill 512 has now been filed. House Bill 13 is the identical bill. In the state of Florida, you must have a bill in each chamber. That bill must pass all committees of reference before it goes to the floor. Once it goes to the floor, it must be read and voted on 3 times before it is melded together for final passage and then sent to the governor's desk. This gentleman is a former House member and a former senator. If there is anyone from South Florida here, you may recognize Senator Frank Artilles. Senator Frank Artilles has been working hand in hand with us to come up with good, safe legislation that's not going to put anyone out of business but help protect the patients. This is our Senate sponsor. This is Senator Hudson. As you can tell, he's got a very friendly, sweet smile. He is also in the running to be Senate President Designate. So he's trying to make a name for himself in all fields. Our next sponsor is our House Bill sponsor for House Bill 313 is Byron Donald. And he is obviously very charming, very handsome, young representative. And he's very loyal to our group. There have been some attempts to come through with strike all amendments already. We haven't even made it to the first committee. And they're already trying to draft strike alls to our legislation. And Byron Donald's staff told them no. So that being said, House Bill 313 has been assigned to go to the Health Quality Committee. And the representative that we're going to be working with there is Colleen Burton. She's been around in Florida. We have term limits, so being around for a long time is almost impossible. But she has been in office for 6 years, and she does a wonderful job. So we are looking forward to working with her and getting it through the first committee of reference. Now in the Senate, its first committee reference is health policy, which is Senator Gail Harrell. Now Senator Harrell is a very, very strong healthcare senator. She and her husband both have sort of run a lot of the healthcare fields in South Florida for a very long time, and she's now our chair and is excited about our bill. So the process that we were going through, I was already telling you guys about the passage, but just so you know, when it goes to the governor's desk, he has 3 options. He can veto the bill, he can sign it into law, or he can pass it without signature, which simply means the 15 days that he has to make up his mind of whether he wants to veto the bill or not. If he does not veto it, it becomes law without signature. Okay, important session dates that everyone needs to make note of because these are my battleground dates. So if you need to contact me and you have questions or information, please be aware that if you call me during any of these times, I will not answer the phone, but I will answer text messages and emails. The week of November 4th, obviously on Monday morning, I will be hitting the ground running and working with the committee chairman to get our bill up and potentially heard by November 12th. That being said, that's our next set of committee hearings. Big date for all of us is that on January 10th is a final draft deadline. Guess what? That's when we're going to know how many bills are out there besides ours that is going to do something to the stem cell world. And that can be a wide open game. Understanding the amendatory process. This is possibly the most important part of being a lobbyist and dealing with the Florida legislature specifically. Each piece of legislation is opened up, and people are allowed to attach amendments. You can debate them, you can fight them off, or you can pass them on to it. That means that every bill that affects anything to do with tissue and organ donation, we now have to watch for stem cell amendments because of a piece of legislation that was killed last year, House Bill 65. So that being said, our usual amount of practice is to generally go to the chairman of the committee and call germanity on it, because currently there's not a whole lot of stem cell statutes. So they would have to be adding us into an existing statute, which would be expanding it, which means it would have a germanity issue. So that being said, that's a little trick to getting it through. Good news for all stem cell physicians and manufacturers are that I read. I have OCD. I read everything that is stuck in front of me. So I read every amendment that's filed in the Florida legislature, which some days is 200 pages and some days it's 2,000. So that being said, I read a lot of legislation so that nothing can slip by me, because I'm paranoid and generally think everyone's out to get my client. So that being said, we move on. An example of the type of interference that we are going to be dealing with. Put it right up here for everyone to see. I did not put it in print. For those of you that do not have the packets or did not get a packet when you came through the front door, there are packets in the front that say 2020 session. They have a copy of all the legislation and all the current bills that I am following for the academy to make sure that we're not going to get stabbed in the back. So that being said, these were the 3 proposals that were brought forward before our bill even fully made it out of bill drafting. We had a bill in the House, but at that time not in the Senate. They were inquiring who our Senate sponsor was going to be. They wanted to know from me, but I don't share information. So that being said, I share the information with the clients and the team that I'm working with, and that's where we are. Currently, this is what we are looking at them doing. To my knowledge, I don't think there's a whole lot of support for the first or the second, but the third is a possibility. So, but that being said, they requested a strike all to do this, which is basically placing House Bill 65 into our bill. So a strike all would mean that they would strike everything after the enacting clause, do a title amendment, and then put this language. And basically, it's House Bill 65 from last year all over again. And Byron Donald's turned them down. So that being said, that's where we're at. We're already fighting early. Okay, this is just a little bit of information in regards to the legislation that is currently filed. I went through page by page so that I could go over with you guys what it did. Basically, page 1 is nothing more than the title page. It tells you whether we're creating or deleting new statutes. Page 2 of the legislation creates the new statutes, very specifically, that creates a stem cell bank registration for manufacturers in the state of Florida, because currently somebody made a comment about Hialeah and people's garages. This is to do away with that and protect it. Page 3 of the bill is a basic requirement that stem cells can only be compounded or administered by authorized individuals. So, and that's a minimum standard that I think everyone legitimate within the industry already does, but this actually puts it in as a safety requirement. Page 4 authorizes stem cell banks in Florida and requires registration. This legitimizes stem cells in Florida. That's what it does, and that's its intent and purpose. And page 5, the most important part of that is it is going to be it's going to require that anything that the Department of Health does is going to have to be handled by an independent third-party panel, and that panel will be required to be made up of actual manufacturers and stem cell physicians and doctors with regenerative medicine. So that is the end of my slideshow. Does anyone have a question about politics?
Thank you, Lisa. At the end of the day, our goal, I'm going to scoot behind you. At the end of the day, our primary goal is to accelerate the availability of both safe and effective therapies for patients in need. And I'm so delighted, Victor O'Brien has joined us today. I'm so delighted that you have agreed to make remarks and offer your insights from a patient perspective.
Got a pack for our third stem cell trial. I'm pretty excited about getting anything back. Anything back is a plus. And I, the last 2 times, got some senses back, and I hope to get some motor neurons back. That'd be really nice. My first doctor said that I wasn't going to walk ever again, and I even read his papers. He says he's talking about me. He says he has high hopes, but in all reality, he's not going to walk again. Day one, 17 years ago, I've always tried to do something to get back on my feet. And along the way, playing basketball, riding marathons on my hand cycle, whatever it took to keep in shape and to try to get some of my feelings back. And over the years, I've received some of that, but it's nice. The stem cell has done a lot, and like I said, I hope to get some motor function back so that I can actually move and eventually walk and stand up to my beautiful wife. And I can't wait for that. Well, yesterday I was hooking up my trailer, and you back up, and you get out of the truck, and you check, and you're off. You get back in, you check, and you're off, and there's trial and error going on, and it's kind of the same thing that the doctor personnel are facing. They try something, they try it, they get results. They come back and try to make it better, get results. It kind of seems like we're doing the same thing. If, let's say, we do this trial and it didn't work, well, they would know what not to do, or they would know to change things. And it's nice to go through this trial to learn, for the medical field, to learn what does work and what doesn't work, and so that they can perfect their methods and help everybody, And all of my dreams are, I'm not in a wheelchair, and I feel like that's my subconscious saying that I'm not going to be in a wheelchair. I'm going to overcome this, and it's kind of amazing that just something that little kind of encourages you and helps you to keep going, and it kind of gives you hope. Even you have that dream that you're walking, and you wake up and yeah, I'm gonna walk. I'm gonna walk.
Thank you very much. I first of all would like to thank everybody here for their time and effort to push science forward. I'm a high school science teacher, and I understand the benefit of collecting data and taking that data and moving science forward. Over the last 17 years being in a chair, first of all, if you asked me if I was going to be in a chair for 17 years, I'd say no. But it's been a long journey, and I am looking forward, and I've always been looking forward for the advancement in the medical, or that one of these days I always believed that I would walk, and I still believe I will walk again, but only if we have people like you that are pushing science forward, and I really appreciate that, and it's because of you that my goal is I'm going to walk. I am going to walk. Thank you.
Thank you, Victor. Thank you for that. To round out our safety panel, I'm so pleased to introduce our next speaker, who's no stranger to you, Dr. A.J. Farshian, who is the founder of the American Academy of Stem Cell Physicians. And Dr. Farshian is going to be the closer for our safety panel. So,
Thank you, Janet. First of all, I wanted to personally thank you for coming all the way from DC. The Alliance for Stem Cell Now, they did a great job. They brought some of the finest scientists, physicians, they put them together. And we got an opportunity to talk to them. And this lady, Janet, she deserves this award, Stem Cell Advocate of the Year, Stem Cell Patient Advocate. Thank you very much. Thank you. So let's get our talk started. I want to talk a little about history of stem cells, the way that the history of stem cell, interestingly enough, it all starts for when Hiroshima fell, a lot of people died that day. A lot more people died after the war from the radiation poisoning. This was brand new to the scientists at the time, so they were baffled by it. So they started to opening and dissecting people. They started dissecting people's bone marrow, and they noticed everybody's bone marrow is dead. D-E-D, dead. Okay, so they started to radiate their rats and mice and giving them bone marrow back, and they saw really great results. In 1961, the Canadian team of Dr. Ernest McLeish and Dr. Till were the first one who described the properties of stem cell. 62, Jonathan Gurdon, the first one who cloned a frog. Now, I was born in 1962, which makes me exactly 40 years old. But you see, 57 years has passed since the discovery of stem cell. When penicillin was discovered, it only took 14 years before doctors started to use it Why is this taking so long? Why are we still talking about stem cells? But first I thought it was the big pharma. There was no big pharma back in 1940s, but actually we do need to know more about stem cells. In 67, they did the first successful bone marrow transplant. 78, they discovered that umbilical cord blood contains stem cells. In the 1990s was the years of progress, all right? This was when they were able to clone the first mammal. This was what gravitated me to stem cells. I was finishing my residency here, and my attending pulled me aside and says, listen, man, if we can clone a mammal, so we can clone a human. So I started to get into stem cell, learning about it. This is a great course. Anybody who wants to learn more about regenerative medicine. I started my regenerative stem cell therapies in 2000, thinking that I'm going to be cloning people soon, but President Bush's administration slowed down things. We fell behind from every country in the world, all the third world countries, including Iran, Pakistan, India, Philippines, Bangladesh, got ahead of us. So that kind of slowed everything down in US. I did the first stem cell therapy on a patient's knee in 2005. This was the first case of a bad actor from academia. In 2006, the guy forged all his documents. 2009, Obama says, okay, the doors are open. Get out there, excel, we're falling behind. But the paranoia sets in. Also, he opened the doors wide open, so the good, the bad, and the ugly walked in together. In 2014, we started to do the neuroregenesis treatments. In 2015, 3 people went blind by a laboratory, not by a doctor. A doctor made these people blind, but a laboratory did that. 2017, we got involved. We wanted to see the safety. So we started the treatments that had regenerative effect. And FDA got involved, tightening some of these rules, and finally slowing some of these people, the cowboys. American Academy of Stem Cell Physicians was conceived back then, all right? Concerned about the safety of America, bunch of us got together and we put the academy together. The academy was set just like a university, a non-profit organization. We saw in 2018 more controversy, more fraudulent activity from academia people, this time from Piero and Becerra, from Harvard and Hopkins. And then there was House Bill 65 that we got the lobbyists, and we went back and forth with Dr. Kit Marsh and trying to fine-tune some of these regulations. And finally, some good news in 2019. About the FDA regulation, you heard it from the man, Dr. Marks. He's got a big, he's got a big responsibility. There is, as I said, when they opened the door, a lot of bad people walked in here. So it's really overwhelming. Like he said, they just don't have enough people. Everybody keeps opening laboratories every day. And when Obama again opened the door for everybody, the good, bad, ugly, you got people who were convicted, you got people, conmen from New York City, you got jokers, you got doctors who lost their licenses, they did Medicare fraud. You get people who go online, they go on eBay, they buy a centrifuge for $6,000, no medical background, no biology background, they open a lab, they're in business. FDA is in charge of people's safety, so that's why American Academy is sitting down and now discussing, going over some of these regulations, telling them who's the bad actors, all right? Are we telling on each other? Yes. But are we breaking the code? No. We got to preserve this field. Next slide, please. So we came up with some of these safety guidelines, recommendations. If you are getting a product, make sure you talk to the chief science officer. Don't just buy anything because the salesman is good. They don't know nothing, okay? Find out how many passages they cultured it. There's a guy who's selling cancer to everybody. He wants to export it to South Florida, to South America right now. He does so many generations. Make sure they had no adverse effect. Make sure, how do they sterilize this? How do they ship their product? Next slide, please. Okay, so we are open to the MDs, DOs, NDs, chiropractors, podiatrists. As long as you're a physician in good standing and you're paying your taxes, you're welcome here. Now, why is that? Because stem cells are too good not to share. I mean, you can't just say, okay, I want to have it, I just want to have all the MDs do it, nobody else, or the academia only have it. It's just too good. You got to let people share it. As long as your state and your board allows you to use it. Here in the state of Florida, they're allowing the physicians of Oriental medicine or acupuncture to give injections. So it's they have the right. You know, you can't deny them the right. So, if that's the case, we might as well teach them to do it correctly. So we can do it ourselves later. So the government doesn't have to restrict everything. Let's play this slide. So American Academy, formerly known as Association, was conceived to make it available. The stem cells share ideas. That's what we are in today. To obtain scientifically sound research, diagnostic tools, and now finally we're initially just a platform for education. We see that we have to do more. We have to get more involved, more proactive, because again, just like the man said, it's too much. All right, everybody, the door's open, so people are just jumping in. So the academy now has delegates. All right, we send our delegates to visit with people. Here we went to Bahamas. We met with their Governor General. There's Barbara Khrushchev, that's part of it. There's Dr. Shea, there's Dr. Shwani. That's the governor. We found out that these guys have the same problems that we have, all right? That was your company right there, you saw that? So, and we talked to some of the kids, some of the future scientists. So the delegates are really busy. We've requested an audience with the President Trump in the next couple of months, but I'm not sure if he's going to be in the mood. So this is a letter that we send to everybody who's a bad actor, who we feel they're a bad actor. The letter basically says, we've been monitoring the way you advertise. We don't like it. The FDA is not going to like it. Your board is not going to like it. You have a certain amount of time to change it and get better. If you don't, we're going to permanently put this letter on internet. Are we telling on each other? Yes. Are we breaking the code? No. We're just preserving the science. Okay, this is the research consortium. It's really the best thing. All right, it's an academy within the academy. All right. So the idea is a group of physicians who are committed to supporting research and validating stem cell therapies. We're dedicated to foster compliance. So basically, we're helping doctors who want to do this. You heard the man. He said that we want people to do it. He said that we just want to do it. We want to do it. Yeah, we don't want anybody else go blind, because bottom line is, when people get sick, it's more work for FDA. All right. So they really want you to do it, but they want you to do it right. We went to Capitol Hill. We met Janet. I was very fortunate to meet her. That's our group. That was like about a month and a half ago. There's Victor. Victor, next time call AAA. All right. So the alliance is a bunch of smart people, really, some are geniuses of our time. There's Peter Marks. There's Dr. Yu. There's Janet. I don't forgot the name of this doctor. But we all had the same goal. We were all concerned about safety. This is a lady in red. She had a child. Child needed stem cells, and she was saying when, where. This lady is with NIH. All in all, it was a great meeting.
The Board of American Academy of Stem Cell Physicians is the official board certifying body of the American Academy of Stem Cell Physicians, AASCP. As a nationally recognized academy with a mission to bring like-minded physicians together to increase awareness and education of regenerative medicine, the AASCP is proud to announce the Fellowship and Diplomat certification. Currently, licensed physicians in great standing will complete 8 modules for certification. At that point, they will qualify to complete a written and oral exam, which is given to them by the 8 workshop leaders. Upon approval, the AASCP will confer upon them the title of Diplomat of the AASCP. And at that point, after 1 year of good standing, they will qualify to become a Fellow of American Academy of Stem Cell Physicians, granting them the title of FAASCP.
So you come in, you do 8 modules, 2 modules every time you come in. You learn about stem cells. You learn the basic science. You learn the literature behind it. Then you take this written exam, which is very difficult. We had some PhDs taking it and they couldn't pass it. But you know, because we prepare the doctors, we keep hammering them. So they were able to pass it. So far, we have like 5 physicians who passed the written test. And then once you pass the written test, you have to take an oral test. And once you pass the oral test, if you are in a good standing, if you've done 3 years of residency, you become a diplomat. You're in good standing, then you can become a fellow after a year. If you're done only 1 year residency, you become an associate diplomat. So again, about Research Consortium, take this seriously. Is committed to supporting research to validate regenerative therapies and dedicated to fostering a culture of compliance within the industry. This is a 361, 351. I'm not going to go over it. Dr. Alexson did a great job. Dr. Marks talked about it. Next, please. Okay, so if you want to go, if you have a product and you want to take it all the way to get a biologic license, you got to go through all the steps, right? And it's expensive. Pre-IND meeting, IND submission, clinical trials, animal toxicology studies, and finally, if you have a good product that is safe, of course, like he said, they will help you. They will help. They will get on the phone and they guide you. They're not after you. All right? They just want to do their job properly. This is very confusing. I'm going to leave that to Dr. Marks and Dr. Alexson. That's all I have for you today.
Thank you, Dr. Farshian. Okay, questions. Maybe we could go with the gentleman over here first. And okay.
Thank you so much. My real question is, what's the practice of medicine versus producing something for that would need some type of PMA? And what I'm asking specifically, let's say, for example, in plastic surgery, you can take fat out and transfer it to another part of the body, and that's, I suppose, considered homologous. But what if we take fat and transfer it into the knee, for example, to use as cushion because it contains collagen, and use it for some type of modality like that? Is that a considered same-day surgical procedure within, the practice of medicine, or is that something else?
Julie, do you want to take, or Dr. Marks, are you still on the line? He might have had to drop off. Do others want to?
What you're describing is non-homologous use, because you're taking it from one and putting it into a different area. So if you read clearly the same day surgical procedure, it would be a non-homologous use. So, but to be honest, I think what you should take the specifics of that and take it to the TRG, because there may be nuances that we're not clear on, and that's why it's hard to answer those questions, because there's a lot of variables in your application that we might not be speaking of. And so in that document, you would provide all that information to them, and they have a group of people, a large group of people that make that decision, and it comes back to you in black and white. So you can ask a bunch of people, is it this, is it that? Some might be very clear, but to be able to make sure that you're getting the right answer, I would say that the TRG would be the best way to go, because you want to make sure that you're compliant when the date becomes for enforcement in November.
Julie, I'm thinking that this is probably a pretty common question, because there are a lot of folks. So I wonder, and I think Dr. Marks had to drop off. I mean, Julie shared, so one thing to think about, Julie shared the article that Dr. Gottlieb and Dr. Marks had. Was it the New England Journal? And, basically, multi-center trial. I mean, this could work, because we've talked with other physicians about this. Multi-center trial, collect data in a registry, and then share resources, because it's pretty expensive. You got to hire lots of compliance lawyer, legal people, to maybe pursue a blanket IND, possibly, or a common BLA. You'd have to use common protocol, collect data, but
Yeah, I think that's where I think that we should go, is figure out how to work together, because there are those opportunities, and I think that there's a lot of you here that probably have a lot of data that could be presented to move you to even submit to the IND, because I know a lot of people say to me, oh, look at all of the effort that you have to take, but you've all done, I'm sure there's a fair number of people that have done a lot of work in here already, and so I feel like that work could be presented, or as Janet's saying, is that you come together to work together with your data, but you have to share that common protocol. I mean, I don't know how many people have IP on their protocols, or if it's something that you can share, if it's an adipose-derived cell that's not unique, or you don't have IP, then it doesn't seem like a problem to be able to share that.
I was just going to add something, and I had a discussion with this as well. Some regulations were written years ago. And as you know, the stem cells is just evolving with the speed of light. So in particular about amniotic fluid, for example, being 351, it is so minimally manipulatively made, it's just you spin it. So maybe some of these regulations going to be revisited prior to November 2020 to just because the field is advancing, and some of these regulations would slow down the advancement. I think I mentioned to you that we fell behind from the other countries, but I feel right now we've caught up and we possibly gotten ahead, and that's because this is America. The way Constitution is written is set up to advance people.
I think what would be an easy way, because that's something that you're talking about that might not require a 351. So what's an easy way to be able to do that and share that with the people in the academy? I mean, I think that would be really important.
His line went dead, and he's trying to call in, so they're trying to get him back on, and maybe he can help with that question.
Okay, yeah, that would be good.
He'll let us know. And while let's do some questions while we're waiting for him to get back on, and we can repeat that question when he gets back on, because I know a lot of people have the same question. I think Phil, and then
for Anthony, for your age-related disease study, it's an IND, correct? So there's a lot of considerations: age-related diseases, joints, brain, organs. So are you doing injections in the joints that have degeneration as well as like an IV for an anti-aging systemic effect? What's your delivery? Because there's so many aspects of degeneration. Yeah. And are you allowed to treat patients that are not in the study with those protocols? Yeah.
Yeah, so that's a great question. That's great questions. And for sake of time and to keep everyone awake, I didn't go through all the nitty gritty of our protocols. It depends on the indication. Okay. So the stuff under IND, we have, multiple things to consider. We're a small company with limited resources. We are doing legitimate science, which is why we can get funding agencies to help support it. But we do have to be very conscientious about what we're doing. For all of our studies, the clinical studies under the INDs, we have pretty tight enrollment criteria. And the reason for that is what we want to do is to decrease the baseline variability, which effectively powers the study better, which means with a smaller patient enrollment, you can get to a statistically significant change or effect that you can see. So these are things that we have to think about. So, I would say on average, somewhere between 50 to 70% of the patients that want to get on the trials because they fail for whatever reasons. Is it working at all? And for me, this has been kind of a almost ethical dilemma, because especially with the Alzheimer's trial, I mean, we have very strict criteria on that, mild AD, with a lot of other exclusionary criteria, because we in the end, if we just conduct a study, we let everyone in and we can't see any significance, it was a waste of time, and that's actually going to delay bringing something to market eventually, because you have to show that it works, and that's the end goal here. Give this therapy to everyone. So that's some part of the reason why we open up this Bahamas registry trial, and that's with the Bahamas Ministry of Health, and it's with FDA approval too. We tell them ahead of time what we're doing, and the reason for that is if people can't get on our trials, here's another option for them. And they can go to enroll in this registry program. We're grabbing real world data from that. It's not as we don't have the high stringency in terms of the enrollment criteria, when we try to get follow-up data from the patients as well. So those are the things. And to get back to your other question, depends on the delivery route. For something like aging frailty and even Alzheimer's disease, that's an infusion, and these are more in terms of aging frailty, this is a diffuse thing. We want the cells to go there. In terms of Alzheimer's disease, the reason for that is based on all the preclinical data, the MSCs are chemoattracted to sites of inflammation and damage, and in a case like Alzheimer's disease, well, you want them getting to the areas of plaques and tangles and where you have the damage, which is a high pro-inflammatory area. And by giving it systemically, we think that they will be chemoattracted to those.
I know we've got a long queue. We're going to go here, then here, then there, then there. And then when Dr. Marks gets on, we're going to go back to you and the follow-up.
Dr. Farshi and I were talking about a company who was replicating MSCs too many times in the lab, and they're becoming tumorigenic. I was wondering, when you get MSCs from bone marrow, do you replicate it in the lab? And how many divisions are you allowing?
So it's a great question, and it's a question she could answer too, because it's a really good question. So there's two aspects to this. One is, MSCs, it's a bit of a misnomer. They're not really stem cells because they do not undergo induction, but they're multipotent cells in the sense that they can it's differentiated into all different tissue. That being said, with any cell type, that terminally, basically they replicate for a finite number of divisions in senesce. We, without giving away IP, we do not bring them way out in passage because that's where you don't want to be. As the cells continue to get further and further passage, they start to enter that senescent state, in which case the cells themselves are starting to, instead of putting out the good molecules, they're starting to put out a lot of nasty things as well that in turn can promote senescence of neighbors.
Yeah. No, I'll just say, so if you're producing a master cell bank from bone marrow or other cells, you're required to do karyotypic analysis, and there's other studies that we do as well looking at the senescence. So you want to make sure you're backing up several passages to confirm your end of production. So that shouldn't happen if you're using a cell source that's a master cell bank.
Thank you. Hello? Hi. I just need some clarity, and I just have some confusion, and maybe it's because I'm a dentist. I don't know. So as far as I understand, and this is for you or for Janet. So come November 2020, we need to use a lab that has an FDA, a 351, if I want to use a umbilical cord biologic. Yes? Wait, let me ask the questions first, and then you can tell me, okay? Okay, I understand I need to practice within my license as a dentist, right? How do I know my state board even can address this question? How do I know that New York State Board is equipped to address what I want to do in my practice? The third question is, I either need to work under an IRB or be sponsored by somebody who has an IRB? Is that how it works?
If it's more than minimally manipulated and it's a 351, you'll have to get an IND to be able to use it. So if it requires pre-market approval. So, well, if someone else is producing your product, which Anthony talked about, you're going to work with the manufacturer. Right, but they won't have your clinical indication. It would be your clinical indication. They'd be manufacturing the product, and they may already have an IND that they can cross-reference. So someone's making a cell, it's under an IND, and you get an agreement where you're going to cross-reference their IND, but you also need approval for your clinical indication. You would also need an IRB. You'd also need.
We're going to go, this is our workaround. We're going to use the cell phone and Dr. Mike, and we're going to use Mike. Do you want him to answer the question? No. Are we good on this last question? Okay. Julie, do you want to characterize it? Dr. Marks, can you hear us?
Yes, I can hear you fine.
Okay. Julie's going to re-ask the question.
Oh, so a gentleman was asking how he could use cells that would be a 350. And we talked a little bit about being able to use another manufacturer. If there was an agreement that could be, if they had an IND, they could cross-reference it, but they would need their IND, approved clinical indication, and he's asking about an IRB. So we said you would need both the IRB and the IND.
That sounds right, yeah, but I think you answered it very right. In your talk, you said exactly the right thing, which is that you're allowed to cross-reference things, and so if you're using a product for an application, you'll have the clinical protocol that you're using, which can be submitted to the Investigation of New Drug application. The Investigation of New Drug application can cross-reference in its entirety someone else's manufacturing procedure, so that your IND may only have to have the clinical protocol that you're using there won't have to be an incredible amount of information in there other than the clinical protocol and some administrative sections. So, I mean, I think that's part of what could streamline things for people.
Okay, one more question.
I'm going to grab, we're passing you around the phone. Hold on. This gentleman has a question. Here.
Thank you so much for helping us. And my question is really what connotes the practice of medicine in a same-day surgical procedure? Meaning that, let's say if we are transplanting fat to a knee and fat has pericytes and preadipocytes that are chondrogenic, is that a homologous use, or, for example, the use of PRP or bone marrow injected into various areas for their biological activity when we're producing it from the patient and then transplanting it? Is this homologous use, or is this falling under 351 or 361?
Yeah, so this gets complicated because certain things, PRP is not considered an HCTP, and it's under a different protocol, and we tend to let autologous PRP, we try to let people do what they are going to do with it. So that's less of an issue for just plain PRP. When it comes to adipose tissue into the knee, that gets to be a non-homologous use generally. But before you take that as the gospel, this is one where I would come in and ask us about it through the TRIP program, just so that you make sure that before you launch down submitting an IND and everything, that it really does require that. So there can be some complexities here, but the honest answer is, in general, if you're taking fat and putting it into a knee, that does trigger the non-homologous use issue. But before you take that as the word of gospel, just come on in and we're happy to have a conversation. You can just use that TRIP program. You don't have to come on in. You can just send an inquiry.
Dr. Marks, we've got a follow-up question.
You address bone marrow as well, as far as how is that looked at? Because usually everyone assumes that same day, same surgical procedure, that's exempt. If you're taking bone marrow aspirate, injecting it into a knee, what are your feelings regarding that? Again, non-manipulated. I'm sorry.
That gets bone marrow aspirate. Legitimate uses, bone marrow and bone chips, etc., used for orthopedic procedures, so oftentimes that is one where we tend to, again, it could potentially be something if it's not manipulated and it's just used. It has the possibility of being, it has the possibility that it could be a 361 in that setting. On the other hand, there are some types of manipulations that could be done, and some types of uses in certain regions, aside from joints perhaps, where bone marrow would not necessarily be a 361. So again, if there's any question, it would be good to come in and ask us.
We've got a lot of hands up. Let's prioritize those who have questions for Dr. Marks. So keep your hand. Okay, yours.
Hi, thank you. My question is really, trying to represent the patients. So many patients, they'll still find that the cost of these treatments are out of reach unless their insurance pays for it. So what would you suggest, or how would you tell us more about what procedures are considered, or that you would consider that insurance eventually can pay for in the near future or already? Because for a lot of patients, it's just not something they can consider.
So in general, insurance companies cover treatments that are approved by FDA or are considered standard of care, right? So in the case of stem cell therapies, it's a matter of getting some of these across the finish line, I think, so they're FDA-approved products, and then hopefully insurance will cover them. In general, that's how things work. And I think that is probably, or quite possibly, the major impetus for people these products and get the safety and efficacy information so that they actually can get reimbursed by insurance, so that a broad array of people have access aside from those who can just pay out of pocket, which is what's going on right now.
So I'm getting the time check. We've got a handful, we've got 3 or 4 more questions. So should they stay and talk to our speakers? Okay, 2 more questions. This gentleman, and short questions and short answers. This gentleman and this woman have been waiting patiently.
Okay, I'm been in regenerative medicine for 30 years. I'm twice the past president of the American Osteopathic Association of Prolotherapy and Regenerative Medicine. The problem we have in our academy there and the academy I belong to here is that we don't use the same forms, we don't use the same protocols, we don't share them, and we're never going to get enough data to do anything until we as an academy do that.
Thank you. Hold on.
So mine sort of is for both Dr. Marks and for Longeveron. When you are going to the Bahamas and doing this registry to get a broad experience with your product in Longeveron, are you going to be allowed to use that in your FDA IND as evidence? And the part 2 of that question is for the broader group. Were that to be true, those of you that are using your clinical experience as, in essence, a registry, if you could find the commonality of your protocols so that you could combine your data, you have the potential to use your past data, particularly if you publish it. And am I interpreting that right, Dr. Marks?
Can you just say that again? I'm sorry. I didn't catch the end of your question.
So the question is, with all this clinical experience that appears to be in this room, and knowing that there's at least one other IND-based company out there using a registry type of background in some of their safety, if not efficacy, data collection, I'm just curious, is it reasonable for this group to look at their protocols and see if there are some common protocols that groups could combine together their data sets to create the safety data set needed to begin this IND process, so that it'll be easier for everyone to get their INDs at least initiated? Does that make sense to you?
You know, the truth is that you don't even need the safety data set. You need the protocols. But yeah, I mean, I think what I would suggest is the easiest thing to do would be, if you get a group of people together that are interested in working together, we're very much interested in helping. So I would have that group get in touch with us. They can get in touch with me. They can get in touch with our industry Biologics email. We'll be happy to have a meeting to go through what it would take to get to an IND. It's not the bar to get to an IND here is something that we've helped a number of people do over the past month and year, actually, as people have tried to come into compliance. And we're happy to help. I think if you have a group that's willing to work together, that's wonderful, and we're happy. I think it's best to just go through what's needed with that group.
Okay. Yeah, and you know, as coming from both sides of this, you know, we've written a number of INDs, obviously with FDA, a number of other you know, academic centers have manufacturing, but they need someone that can manufacture the cells, and we help them with their INDs. And that's probably the biggest hurdle in terms of this group here, is finding someone making a product that is, you know, done under the blessing of FDA. It's done in a CGMP manufacturing facility under all regulations. You know that what you're going to end up giving the patient in there is it's a good product that is free of contaminants and other things you don't want to give the patient. That's the hardest thing that the manufacturers have, and that's a cross-reference. The IND port, in terms of what you would need, you could work sponsor, the manufacturer, or you can write that yourselves. And what you're really down to at that point is just making a protocol, the ICF, the informed consent form for the patients, and then there's some other minor stuff in there, and there's a lot of options to do that. So the Bahamas registry is for multiple purposes. It allows people to get into our clinical studies that don't necessarily qualify because of whatever reasons to be on the clinical trial itself. So as a basic scientist, I did have I was like, we are using the data, yeah, and it gets reported to FDA. It goes as part of our annual report to FDA. We do what's called a DSIR, where we integrate all the information from all of our domestic and international programs together, and we present it to FDA in an annual report. So yeah, and we are grabbing that evidence. It's yeah. Oh, in the Bahamas, it's a completely open label study, and one of the things that scares me is when people come to our trials and say, if we're not going to get your product, we're going to go to Mexico, Panama. And I'm like, and what are you going to be getting there? I mean, at least here's an option where you're getting something that's being manufactured under regulatory oversight.
We look forward to continuing the conversation. Please join me in, just so we can let Dr. Marks go. It's late. You've hung with us a long time. Please join me in thanking this wonderful panel. Great discussion.